I'm very proud and happy to say a few words about the MS Research Prize, which we give every other year. And the idea, of course, is to promote excellent research in Switzerland. We had already, 2 years ago, we had this prize awarded. Of course, with 100,000, so it's not a trivial amount. And Jens is very knowledgeable about this. And then it's sponsored independently, not by pharma. That's another thing to know. It's not through common donation and no, not pharma. And an important issue is it's not for the new sailboat on the lake for the prize winner, so it's exclusively thought for research, which is self-understood. Press. So I try to slowly lift the fog, as you know, and the prize winner did also importantly lift the fog among them from the blood-brain barrier. If it comes. Well, it's one too much. But the fog is lifted and they grow even vines, as you see. And of course, the prize winner kept going south a little. And in the city she is. And you see here, if you look close enough, already a little bit of a symbol of the blood-brain barrier. So, sorry, I try hard to press. And the winner is, and now the fanfare. Well, I try hard to keep the choreography. Please.
Thank you very much.
So, we have, yeah. Now we have to do the picture.
Exactly.
Yeah. The next.
Everything.
Yeah, the chocolate and the flowers and
the number one.
Yeah, the number one. Yeah, let's put it in the picture. You have to hold. Well. So we can. Let's like this.
Brother.
He wants
we need
more flowers. Yeah.
we are not enough.
You'll have to take this home. Okay.
Thank you.
Very good.
Thank you.
Now, the next step is there will be the talk of the awardee, but now I have the pleasure to invite our special guest, Hartmut Weckerle, whom I introduced already last night, today, and now you see why he is here. He is the long-standing mentor and tutor of Britta. Of course, he is at the Max Planck Institute. He's the director of the neurobiology, and recently he also published on the gut-brain axis also about autoreactive T cell traveling from the gut to the brain. And he is certainly the right person to have the laudatio. And I'm glad to hear that he is also collaborating with our people in this room, like of course Ludwig and also Francesca indicated. So he's very active in this field, and I'm glad to invite you to have the laudatio now.
Mm-hmm. Well, the surprise is over, but our awardee sits here. Now, dear friends and experts in MS research, Today is a great day for our field. The Swiss Society of Multiple Sclerosis is, as you have heard, awarding its biannual prize to a person who, not like very few others, have imprinted experimental autoimmunity as related to multiple sclerosis. And I am obviously very honored to participate, to provide a laudation to our awardee, but I'm also deeply moved because, as you just heard, I do have special connections to our prize winner, Britta. Well, because she has been one of my first female PhD students, and perhaps one of the most successful of them all. Yes, this in a way raises emotions. Now, I would like to start out with her life work, the interest which guided her scientific career. Now, and this is a very old, perhaps one of the oldest dilemmas and problems in multiple sclerosis. It dates back to about 145 years when Charcot observed the structural deficiencies underlying the disease. You see these brown fields in the brain-white matter, plaques, the MS plaques. But perhaps in our context, even more important was the observation of Babinski. Well, you know him all, the gentleman with the two test, who observed that within these pathological lesions, there are pathological cell components, round cells around the blood vessels and disseminating into the surrounding brain parenchyma. This, one could accept, yes, that is the structure of MS plaque, but it creates a dilemma. Here we go, yes. These cells, the disseminating cells, are not of brain origin, but they stream into the organ through the blood vasculature. You see here two of these definite, in the experimental setup, definite pathogenic immune cells, lymphocytes, which break their way through the thin blood-brain barrier. This is actually, one would assume, the initial step of the creation of multiple sclerosis lesions. Now comes the big question. We all are convinced that the blood vessel walls, in contrast to most other vessel walls outside the brain, are tight. They prohibit soluble macromolecules and most circulating immune and hematopoietic cells, their traffic into the surrounding parenchyma. That is dogma, thousandfold confirmed. But now we see that blood-borne T cells or other immune cells have made their way into the parenchyma. How can that be? How could they manage to cross the tight blood-brain barrier? And this is the long-standing dilemma or problem of neuroimmunology. And this also, I would say, is the topic of the life of Britta Engelhardt, allowing her to do research which largely helped to understand these unusual and so important processes. Now, the solution came about 1985, I think, way back in the last millennium, when the door opened in Würzburg, and in came a very young, very committed medical student from the human biological faculty in Marburg, who came in and clearly stated she wants to join us. Why? She's interested in multiple sclerosis, and here that we are doing experimental and clinical work towards the solution of the problem. Well, she came in with super excellent marks. Her behavior convinced me, it was self-assured. And, well, I thought, yeah, she might be some new asset of our group. I, hired her. And this coincided with another thing, equally important. Just around this time, we made contact with a brilliant young biologist, an expert, a world-leading expert in the biology of brain endothelial cells, the vascular wall cells. He could culture them and look at them in vitro. Why shouldn't we join forces? And Britta was very eager to be the person to do the job. What she did is she co-cultured the Riesau type of endothelial cells, organ-typic, with brain autoaggressive T lymphocytes, who were the mainstay in my neuroimmunological group. So how would these two cell types interact with each other? If we find particular behaviors, could they underlie the first stages of the MS plaque? This was basically the task and the successful solution of this work. Now, she gathered data on this interaction and found very interesting, unanticipated data. Enough to be published in, at that time, leading cell biology journal, the Journal of Cell Biology, publication which really made recognition, and in a way, as it turned out, was the first step in Britta's scientific career. Now I would call this episode "A Star Was Born." Forgive me this astronomical metaphor. Now, this was actually the lady. That's how she did not change. Well, perhaps for very keen observers a little bit, but that was she. She was convincing, and she proved her value. I was very happy to have her as a doctor and then afterwards as a postdoc. Now, what followed up? There was a follow-up, obviously, like all very energetic and demanding students. She, undertook a migration round. She got, obviously, with her previous doctoral research, good offers all over the place, I would almost say. And among them, she chose the laboratory in Stanford of Eugene Butcher. He was a person who led the field of cell adhesion, cytokine signals that led immune cells and others to different sites of the body. She was aware that would be helpful to understand the brain endothelial T-cell interactions. Actually, a topic which I understand was not being elaborated in Jean Butcher's department. She introduced a new aspect there. Now she was there, I think, 2 or 3 years, very successful, published a number of good papers, and then somehow she felt like returning to Germany after some, well, minor turbulences. She settled back in the newly founded endothelial department of Werner Riesau, remember him, who unfortunately died very soon thereafter, in Bad Nauheim, a Max Planck Institute. Again, she adapted to the local programs and certainly fertilized the local research until Werner Riesau's demise. And thereafter, together with Dietmar Westweber, she moved to the institute, Max Planck Institute in Münster. Good, but that's not all. She developed a very independent image and was very successful in her field. But then comes the Big Bang. She became, well, unexpectedly, I would say, as a German for Switzerland, she became the chairperson of a top renowned Swiss University Institute. Imagine that. This was 2001. Well, we were all proud of her, regretted that she left us, but it was a good move. The, well, nascent star who was born in Würzburg, well, became a fixed star, as this astronomer teaches, right? And ever since, for the past 25 years or so, she has become a dominant, well, benevolently dominant expert in cerebrovascular biology. And I will not go now into the details. I'm already over my 10 minutes. I would need certainly about 40 minutes, but I won't, because she will give you insight in her present interests and how she came to this situation. But I will simply mention what has coined her success in preclinical research. One thing was she had really a sense of developing and combining very divergent technical approaches to form a coherent research program in vascular immune research. She combined molecular biology, cellular biology with top imaging. Just one example, in vivo experimentation with new transgenic mouse models, and you just name it. She contributed to the field of vascular immune signal molecules, what I mentioned before. She clarified tissue or area differentiating signalings for invasion, and she always had a keen eye on therapeutic application of her research, actually from her work of Jean Butcher, in an indirected way, the development of natalizumab, which is used obviously in MS, has some roots in her work there. Now, she is, of course, a very, very productive researcher, and in our field, this can be measured by the number of papers released from the group and by the citation of, where should I say, competing laboratories or learning laboratories. What you see here, she usually publishes about 20 or more or a bit less of papers per year, and this is cited by, I would say, almost thousands of other scientific colleagues. So this catapults her basically in the top league of this specific research area. Now, but Britta Engelhardt, I really should mention that, is not just a dry experimental biologist, but, well, she has qualities which I now could quote. I simply mention her knowledge of the field, but also her interest in academic administration, in student affairs, but most important also her combination with empathy and interested in the human field of academic debate makes her one of the very, very much sought after members of committees. Presumably, sometimes she may think almost too many offers to accept. So, presentations, how many presentations? Reviewing for journals, for research organizations. She seems to be a very popular teacher. I have not heard it, but I hear it from Bern. And she has been gathering a good number of scientific awards. And I think among these, today's Swiss MS Society's award is one of the most shining. Now, obviously, we are here to congratulate you here for your unique productivity, your developing, your success over the past years. And the prize is also thought to, well, to encourage you, to support, to fuel your research, your joy in research and your success. Now I should conclude, and that is a fact of life, obviously. The prize doesn't come just with honor. No, it also comes with money, real money. Thank you very much.